I just wanted to kind of get things started. I think we're all really excited. This is not going to be a usual panel because you have me as a moderator. It'll be not JPM, but it will be deep, it'll be scientific. We're going to be talking about longevity and the future of medicine. We're not going to be talking about some of the other stuff, which is not medicine or longevity. It's just stuff that people sell online, right? Some of you know what I'm referring to, right? So with that, I'd actually like to let the guests, we have an incredible panel tonight of guests. I'd like to let the guests introduce themselves. Most of them don't need introductions, but if they do and they're too modest, I will call them on it. We have some incredible pioneers here, so maybe I'll hand over to Vijay to do a little introduction on himself.
Hi, thank you all for coming. I'm Vijay Pandey. I'm a journal partner at Andreessen Horowitz and a founder of the BioFund there, as well as a professor at Stanford. My history has been involved in startups my whole life, starting when I was 15 in the first one. What I love about this space is that it reminds me of all the things that I love in tech in terms of innovation and ability to really change the world. But, brought into biology and healthcare, I think the ability to rethink the way we think about healthcare with longevity, I think is super exciting, and I'm glad to try to be a part of it.
Hi everybody, I'm Kristen. I'm the founder and CEO of BioAge. We're a startup company focusing on accelerating drug discovery for aging. In particular, we take a data-driven approach to find the molecular signatures that drive human aging. So I'm really excited to be here tonight and to have this great group of people bringing together pharma and tech. So looking forward. So I started BioAge a couple years ago because evaluating a therapy, well, first of all, there's this enormous potential in aging, right? It's been estimated that if you have a drug that cures all cancers, that will add just 4 years on average to lifespan, because as you get older, all these chronic diseases, that your chance of getting them and your chance of dying from them rises exponentially. But we know we've had for over a decade now various things we can put into mice that delay the onset of all these diseases at once. And if we could translate those to humans, they would have a tremendous impact on health, much more so than treating any particular disease at a time. The trouble is, though, the list of things that works is incredibly short. And one of the reasons why, or the reason that I think is sort of the biggest bottleneck, is that testing whether a drug works in humans or in mice for aging takes decades or years, because you have to see if they live longer. So I started BioAge to build a proxy. Like, what if we could measure something in your blood? What if we had a true biomarker of longevity that could accelerate the cycle? And we could iterate a lot faster and find a lot more molecules that could increase lifespan.
So theoretically, that basically means it may be possible to go past even the limit of what we now perceive, right? As when we die. Is that right?
Theory and practice, Ryan, but...
Theory. I'm pushing theory. A lot of scientists want to be very hyper-realistic, which is good. But one of the things that I've tried to do is to get scientists to dream. What if, right? What if it works, right? And so...
you could speak to aspirationally, what...
Yeah, aspirationally, we know there are mammals that live well over 200 years, right? There's nothing in biology that says that isn't possible. I think it's going to take a long time to get there, but I feel that gains of a couple of decades are actually going to happen fairly quickly.
I like that.
Hi everyone, I'm Laura Deming. I run the Longevity Fund. Started about 6 years ago. It's one of the first venture funds to focus solely on investing in aging technologies. And I got interested in space about 10 years ago and have been just kind of burrowing at it ever since.
And when were you first in the lab, Laura?
So I actually first started when I was 12 in Cynthia Kenyon's lab. And I think one of the fascinating things to me was that it wouldn't have been possible to be interested in this space earlier than that. Like the first genetic mutation that we ever found that impacts worm. Oh, sorry. It's like Ryan said not to do too many details.
Good.
Really quick.
Get it exciting. That's what we want.
No, but the first ever genetic mutation that we found that increases lifespan in any organism tested was in 1983, approximately. And it was only validated in 1993, which is a year before I was born. So, realistically, you probably would have been irrational to be excited about this space before that. But now is kind of a great time, hypothetically speaking, or hopefully speaking, at least to be sort of in the space and excited about it.
Awesome, I like that. Hi, Tom.
Hi, everyone. My name is Tom Rando. I'm a professor at Stanford, and we work in my lab on the biology of aging. And like many researchers in aging, I didn't enter science with an interest in aging, but I got drawn into it by the research we were doing, and a lot of our work has focused our attention on the possibility that there are factors in the blood that drive aging, and therefore maybe factors in blood that can delay or even reverse aging. So a lot of our interest is understanding how cells and tissues age and whether it's possible, in fact, to reverse that process. So we do this in the lab. I'm very interested in translating a lot of what we do into the commercial space. I'm very happy to be here.
Awesome, thank you, Tom. And just a reminder for everyone, we like Facebook, we like Twitter, we like Instagram. Feel free to take pictures, tweet, live stream anything you want. It's very interactive. If you feel really inspired by something, shout it out. But the format that we're actually going to follow is we're going to say about 20 minutes of conversational Q&A up front. And then we're going to open it up so that you'll all have an opportunity to ask a lot of these incredible panelists questions that are burning in your mind. But just to really get us started, because we have a real incredible mix of tech venture capitalists, investors from the biotech space, builders, founders, scientists here in the audience. So maybe if we just start at ground zero, which is really this whole longevity thing, right? So is this real? Is this something that we can make real within our lifetimes? Is this something we can actually turn into therapeutics? How is this the future of medicine? Maybe Tom or Laura, you guys want to? Tom, you want to?
So when you say the whole longevity thing, you mean living longer?
Living longer.
Living longer.
Yes.
So
Yeah.
Interestingly, clearly over evolutionary time and then over modern time, lifespan has increased, obviously, and but that's obviously average lifespan. So the average time people live has increased dramatically, and at the same time, we really don't know too much about what's happened to maximum lifespan. There's sort of a hint that it's been gradually increasing, but it's been minuscule compared to how average lifespan has increased. And so that really raises the question about what are we really interested in, or what two things are we interested in? One is increasing maximum lifespan. Can we live to be 120, 140, 160? But really, and a lot of our work, I would say, focuses also on the idea of living healthy. And so as lifespan has increased, there's really been an ongoing debate as to whether we have actually shortened the period of time at the end of our life when we're not healthy. And we'd like to think that, but that's an ongoing debate. So I think one of the big challenges and how I even define this is two things. Is the idea of what is the maximum lifespan, but even more importantly in the short term, an achievable goal is, by the old expression, compressing morbidity, so that we have a lot more of our life that we live healthy and a lot less of our lives that we live unhealthy. So I think those are two aspects of longevity that I think about.
And Tom, I just want to chime in. You are not just a theoretician on this, you're a practitioner. You really pioneered
I live
a lot
longer?
Yes, well, you pioneered a lot of the work on parabiosis, and maybe if you could just tell the audience what parabiosis is and maybe some of the outcomes.
Okay, so parabiosis is this weird experiment whereby you connect two animals together and they develop a single shared circulatory system like conjoined twins over the course of a couple of weeks. And then they can live like that. And we do this with mice. And the experiment we did now, 15 years ago, was to connect a young mouse to an old mouse and leave them that way for a couple of months. And then, and then other people have reproduced this in other tissues. When we go back to look at their cells, the old cells, the cells from the old animal have, in a sense, by the criteria that we measure, look like they've become younger. Now, there's no free lunch, so the cells from the young animal look like they've become older. So this is really what has driven our thinking about what's in the blood that might drive this. And so this really relates to kind of the idea of developing therapies from this that can change health span. So that is not so much about lifespan. We don't really know what happens to the lifespan of these mice, but it looks like we can change the health of the tissues.
Fascinating. And so the question is, why now, right? Humanity has been, since our inception, trying to look for the fountain of youth. And I don't know if you guys wanted to maybe chime in, Kristen and Vijay.
I think that we have the science now and we didn't before. Like to Laura's point, the very first mutation that reproducibly made worms, not a mammal, live longer. That was not that long ago, right? That was a couple decades ago. And in the next decade after that, there became a very long list of things that could make these invertebrate species live longer. And sort of handful of things that work in mice were discovered pretty recently. So we're really, there are already things that work, and we're already sort of in the, I think, the very early days of mapping out the space. There's going to be a lot of other things that work, gene mutations and small molecules and other interventions that work in mammals.
Yeah, and the point I would add to that is that when we're talking about longevity, often we're talking about extending lifespan, but to me, really, this is the future of mainstream health. Maybe one way to sort of frame this is that if you think about the early things that we've done as a civilization to extend health, it wasn't that we were getting dysentery and we were getting microbes and then we came up with all these antibiotics to treat the issues after we got them. No, we came up with things like infrastructure for sanitation such that we never had these problems in the first place. And I think this has that sort of style in my mind of instead of fixing problems as they come, which is just this constant nightmare, the ability to just never have them and push things back is the type of fundamental advance that would be akin to sanitation, akin to the great advances that civilization has come up with.
And Vijay, in terms of an oh wow moment, I guess this is for you and for Laura, you know, which companies and which technologies have given you both kind of those oh wow moments where we're getting there, right?
Yeah, well, I mean, and so I'm very biased because, you know, on the stage here we've got Tom's experiments. For me, the parabiosis experiments was the key oh wow moment because the question wasn't could longevity occur and could a youthful phenotype become engendered? The question is, what is it in the blood of the younger mice that did things that would engender it? There was a causality there. And then, and I should mention, we've invested in Kristen's company, and with Kristen's company and also Tom's work to try to understand what is it and how that could be used as a biomarker and ideally as a future as a therapeutic. Those oh wow moments, I think, is a really pivotal moment in the last few years.
And Laura, in terms of actually putting, you know, some numbers on this, right? Because I think when we talk about longevity, we often talk about, well, you know, we can extend healthy human lifespan or extend healthy lifespan. What are the actual numbers that we know, at least in animal models?
Yeah, so I think one of the funny things that was an oh wow moment for me, at least, coming into this space, was I was like, all right, I have to do a very comprehensive overview. Like, what are all the things that make mice live longer? And when I started, I remember thinking there's probably like 6. Like probably there's maybe 6 things and maybe we get like a max 10% increase and that would be amazing. And this was a long time ago, I think 6 years ago or something. And it ended up being like 95. And I would say most of those things probably aren't reproducible. Like a lot of the studies were with very small numbers of mice, there are lots of confounding factors. But the fact that there were so many and that if you looked at things that were actually in the clinic, a lot of drugs that were being developed for things like diabetes, if you gave them to mice, produced an increase in lifespan. I mean, that to me was shocking. Like, either it's all incorrect science and none of these studies will reproduce, or this stuff is a lot more prevalent than we think, and we just haven't been looking for it for that long. Numerically, I would say we have achieved a maximum of about 68% increase in lifespan with single mutation. And if you think about that for a human, that's actually an enormously large increase for a single genetic mutation and one experiment in like 1996 that's never really been fully fleshed out. I mean, how much longer could we get that increase to be? We don't really know. So I think we're just beginning, and that's part of the excitement of working in this field, at least for me.
So that's a fascinating implication. One mutation, 68% life extension, and these things may be independent, right? Like these.
I should point out that's a dwarf mouse. So, like, not that you would want this mutation for yourselves.
It's a great time to be a dwarf mouse.
Yeah.
Right. Yeah.
Great time. So in terms of you know things that we can do today in terms of prevention, I know that that's something that's on people's minds. Have you all been looking at potential interventions or ways that we can actually prevent or slow down some of these aspects of aging? What's most interesting, most promising to all of you?
Well, you know, since no one else is jumping out, I'll just give maybe a framework to think about it. You know, there's different ways to engender longevity. I think one way would be to follow along Tom's experiments and try to think about what is it that we can do to help our own bodies engender longevity and to be healthy. Another model, which I think is radically different, is to think about, like, you know, my parents keep their cars forever. I drove a 1974 Dodge Dart for like way too long, and the reason why is that, you know, they kept on swapping out parts, and that you could swap out parts, and so those are very different types of models. One where your body's sort of doing it itself, and the other where we're doing it with help. They're not mutually exclusive, and there may even be other models. And so, in terms of answering the question, I mean, there's many different ways to get at it.
And that leads to the question why we die, right? Why we die and how we can fix those pieces that kill us. Tom, it sounded like you had a thought there, reflection.
So, you mean, so what are the causes of death? What are the causes of adult death?
Yes.
Yeah, I mean, so if you look at the causes of adult death in, today in the US, it's a pretty well-known list. So there's heart disease and stroke and lung disease, kidney disease, accidents, suicide actually is high. But, many of them are just the diseases of aging, right? That's by far and away what causes adults to die in this country. And the kind of driving mantra of the biogerontology field is this idea that if one can understand what it is about aging that leads to a susceptibility to all these diseases, and if you could begin to target aging, you would cure, so Kristen was saying before, you would cure all the diseases, or at least you would treat, prevent, delay all of the diseases by targeting this basic biology of aging. So yes, as Vijay was saying, you could kind of one at a time start to replace things or start to treat diseases, but that's the whack-a-mole approach. You get one disease, you treat it, and then they live a little longer, you get another one, you treat that. That's doable, that is what we do. But the promise, in a way, of this emerging, developing field of the biology of aging is that you can think about the causes of death as being caused by one thing. And that there's a kind of single common pathway that leads to susceptibility, and that's what you want to modify.
That's a perfect lead-in to my next question. So let's assume we find this molecule, this therapeutic, this stem cell therapy. How long is it going to take us to get approved, right? And so this is really a question for Kristen, for Laura. Is this going to take 50 years?
Well, that's the trouble right now, right? If you have an anti-aging drug, I think all the companies working in this space, they're not taking it through in a clinical trial for aging. Then you'd need 20,000 people in 50 years, at least until there are good biomarkers available. We're working on that. But the strategy that people take right now is they just take it to the clinic for a particular disease because, frankly, there are so many diseases where aging is causal. You have your choice of various common or orphan age-related diseases. Then you can sort of get your foot in the door. I mean, really, this is the story of what happened with statins, right? They're basically an aging drug now. If you're over 40, you have some risk factors, you get prescribed a statin. But that's not how they started. They started by being approved for familial hypercholesterolemia, an orphan disease where people had super high levels of cholesterol, like a tiny population.
But a well-defined endpoint.
But a well-defined endpoint, so you could have a fast trial. And then sort of widening over time. So I think that's a strategy we have to take now. I hope that's not always the case. I hope that, with the right biomarkers, we can accelerate that for what we really care about.
And in terms of, looking at as we all age, right? All of us here are aging. There's going to be increasing demand for new types of therapies as we get more debilitated, we get weakened as we age. Is there a kind of a space where you all think that an investment would make a significant difference, an area where it would really accelerate the entire space of aging and longevity therapeutic development? Is there kind of a fulcrum somewhere?
It's just so funny to hear you think like the whole space. Like we
yes,
just need more, like enormously more amounts of capital going into the whole
and
space.
That's not necessarily an accelerator hint, right? But
Actually, I think there is one interesting thing, which is I think, and Kristen, maybe you mentioned a little bit something similar when we were chatting earlier, which is when I first came into the space, my presumption was that we were lacking targets, and that really what was needed was more basic research in aging and more understanding of the biology in order to allow us to then know what to develop drugs for. My current understanding is that actually we have too many targets. Like we're kind of overwhelmed in interesting things that we could be going after. But most of the things that we'd like to actually downregulate or upregulate, we can't physically. And so it would be kind of ironic, but possibly one of the biggest holdups to aging drug development right now is drug development in general being a hard sort of row to hoe, so to speak. Which was an interesting and unexpected conclusion.
This actually hits perfectly, Vijay, your recent controversial Medium post, exciting, popular, and controversial about the spaghetti code of biology.
Yeah, I mean, I think it's not a secret to anyone here that biology is really hard, and that I think, and wasn't, even if there was somehow some intelligent designer, he or she didn't do a very good job, in terms of the mess that's left. And so, I think traditional drug design has all these challenges. I'm sort of at a loss too for like where there would be a magic bullet. I don't think there would be. I think my gut feeling is that a portfolio approach then is the only thing to do, that there's multiple different ways that we can try to go at the problem, and that we'll see which is the most successful one first and go from there. Also, it's very possible that with these multiple targets that there will be cocktails that will be very interesting to hit the targets. And just as SNPs are additive, small molecules could be additive, we'll have that type of more sort of library effect as time goes on.
So if I could just add, and this is a plug for Kristen, I mean, clearly, as with any biomedical research, biomarkers is what really changes the game. But this is clearly true in aging, but you look at any disease. I mean, you look at the trials for Alzheimer's disease, what's the problem? You got to spend a long time and get a lot of people to see an effect. Now, what if you could treat someone with Alzheimer's disease for 2 weeks with something and say, they're getting better? I mean, it would be a huge game changer. Now, so biomarkers are hard. It's true in every disease. The better the biomarker, the faster that really accelerates. So, I think what Kristen doing is critically important, and I hope that there'll be more and more investment in that idea of how do you accelerate the process of understanding if something's working in this strange space of therapies for aging.
I'm going to add to that because, I completely agree, but not just to evaluate therapies, but even to evaluate subjects, right? Because it's Alzheimer's, it looks like right now we're intervening too late. So it's a matter of finding the right biomarkers. I mean, people, to a clinician right now, present as healthy, but you can still see with the right biomarker that they're already on that path.
It's a lot easier to deal with the house when you have a smoke detector, and before the house fire is burned down, the whole house.
Yeah, and that's the important point I want to make too, is that I guess I think biomarkers are a lot easier than drug discovery. I think that with the technology now and with the right samples, we can build good ones, and that's going to be sort of a key enabling technology.
And I think it's important to note as well, biomarkers are well known and well understood in terms of other therapeutic indications, areas that the FDA currently regulate, and they're very transformative, right? So if you look at an area like orphan drug research, some of the biomarkers that have been identified transform the lives of many people as a result of identifying biomarkers that the FDA approved, right? And that's why it's so important to get the FDA to recognize certain aging or longevity related biomarkers so that they can actually approve regulated and regulate these drugs so that we can speed them to market, right? One hot area that I think is still pretty hot and probably worth talking about is, what's driving the investment in this space? The moment that Google moved in and created Calico, all of a sudden, this space really transformed, right? It became the talk of the town, at least for Silicon Valley. We had a few Boston VCs coming in and really getting excited about the space too. So really, was it the money that got people excited, or is it really the fundamental science that's driving this right now?
Probably, one drives the other, right? So it's hard to know causality.
Yep.
But, the joke is that people thought this was the domain of vampire billionaires, and that now we're realizing that this is really just mainstream health.
I think one of the other interesting things too is if you look at a fund like Arch, which had a recent sort of nice proof of concept with Unity Series B going quite well, they actually had a prior company with my former mentor Cynthia Kenyon, Elixir, that failed previously in around 2006, 2007. And so Bob Nelson's been a true believer for a long time. This isn't a recent phenomenon. I think this tracks nicely with what you see in other similarly sort of frowned upon areas. So CAR T, for example, something that people have tried many times over. I mean, Arch had done Xsite, which had failed as well, doing exactly the same thing that kind of Juno then subsequently came along and succeeded at. And so, yes, there's been a recent wave of interest, but also a lot of these guys have actually been trying and failing and then coming back around for the past couple decades, actually. And it's kind of exhilarating to watch them finally start to pick up momentum based on all that effort.
Go ahead.
Yeah, I want to say too that there definitely is a boom right now, but I think it's important to keep in mind that how many aging companies are there compared to the number of cancer companies out there or diabetes companies? It's still so small, So I think I still think the real bottleneck is in between the discoveries that have already been made and commercializing them. So I think there's a lot of room for it to grow.
I think my own experience in this is that it's almost like a herd mentality shift that I see, because I remember presenting this idea kind of that maybe there are targets and maybe there are therapies for aging about 12 years ago to a couple of companies, and they laughed me out of the room. I mean, and then they said, this will never happen because any treatment would have to be as safe as water. I will never forget that phrase. I mean, that was something they knew very well. I'd never heard that before. And of course, there will never be a drug as safe as water. And I think that, that what has changed, at least in part, is that thinking that, okay, maybe not. Metformin, it's not as safe as water. It's pretty safe. But just the idea that there may be acceptable side effects, as there are for any drug, if you could really get at these fundamental approaches and treat lots of diseases. And again, I just think that's just a change in mentality. And I can't speak for pharma, really now, but Novartis is doing trials. So at least big pharma is getting on board.
It could be that in San Francisco, the quality of water has changed itself.
So maybe just to touch upon, that translational step as well. So how many technologies does the panel really think are ready to translate out of the universities and become actual startups and companies? And do we also, and feel free to answer either side of this, do we need to invest more money in some of the basic research that we're doing in our institutions and universities?
Yeah, I'll jump in. The basic research side, that's an easy one because this is so early that, and the level of basic research in general has been, in a relative sense, declining. So that's an easy one, and I think given the significance and frankly just the excitement about the biology, hopefully that would be an easy one. Where I think the challenge is how can you have a company that comes out of there with an interesting technology, how do they get to market? And because death is not an indication. There's no code for death, no code for longevity. So, you have to come out with some indication that would correlate with aging or connect to aging and get past the FDA. And so, coming up with the right go-to-market is part of the challenge.
Okay. So any other thoughts?
Actually, I have one last thought, which was, so something that really struck me when I first entered Cynthia's lab was, if you look at the two canonical papers that came out of her lab in the 1990s, the first one that ever showed a mutational impact on lifespan was done by a rotation student in her lab. It actually wasn't a grad student. Nobody in her lab, which was at that time focused on developmental biology, wanted to take a flyer on this project. And so it was like a 3-author paper with a rotation student. The other thing that I think most people don't know is there was a paper that came out 3 years later that was also foundational, showing an impact, a reproduction on lifespan, that was co-authored by a 14-year-old. It was a 2-author Nature paper that's one of the foundational papers in the field of reproductive impact on aging biology. And nobody else, again, in her lab, even after the first paper made this huge impact, wanted to take a flyer on this new problem. So from my perspective, there is a question about basic research funding. There's also a question, where are these odd people who are willing to take flyers, and people who aren't putting their careers at risk and feel like they have the impetus? And one of the questions for me has been, is it a lack of those people that's what's holding the field back? Not so much a lack of total funding or grad students willing to work on this space in general.
I just wanted to interject, and it is still the case though that everyone knows probably in this room that funding for basic research is at an all-time low right now, but what you might not know is that it's particularly awful for aging. So aging is just a tiny, tiny slice of that. So even within academia, there's just not that many people working on it. I did my PhD at the University of Toronto, that's Canada's largest research university. There were 0 labs that worked on aging. And when I came to Stanford for my postdoc, there's a few, like Tom and Stuart and Anne, and there's probably, 10x the number who focus on cancer alone, right? And the funding reflects that. And that's sort of the very first step of the pipeline. And I think that it can be, I think that could be really transformative for the field. I think,
Great, so we're moving into our final lightning round. So anything you'd like to leave the audience with, and then we're going to flip to all of you for the questions, right? So any kind of parting thoughts, parting words that you'd like to leave the audience with?
Well, I'll just give one parting thought, which is not biology, but which always comes up, something we talk about a lot, which will be, if we're successful, what will the societal implications be, and who will pay for it, and who will get it, and this will end up being an important discussion to make sure if there are developments as something so fundamental as aging that it's available to everybody. Who's going to pay for it? That's the question. The optimistic view is this will pay for itself. It will result in decreasing costs of diseases of aging. And so the cost of providing it will pay for it. That's the optimistic view. Of course, the pessimistic view is not that. And so I just throw that out as a food for thought, as beyond the biology, beyond the therapeutics, there will be societal questions, there'll be fiscal questions, and there'll be ethical questions.
The challenge there is that you could suggest that the alternative is we just encourage everyone to smoke as much as they can. That would be the cheapest solution.
Any other thoughts? Parting thoughts? No? Okay, so I think we're opening it up. Okay, questions. Oh, okay, let's start front and we'll move back. All right.
Thanks, Ryan. I had a question. Well, actually, I have so many questions, but two of them, I think. One, I wonder what, this goes to Ryan's question about what area or what company might break out and attract additional funding. And I've always thought that companies that are developing glucosapine breakers, which glucosapine as you age increases in your body and causes wrinkles and arterial stiffness. So I've always wondered if a company like that would, attract a ton of funding, cosmetics and everything. And then the second question I had for all of you, or any of you, was, so I have a friend, kind of crazy, he's in his 50s, Darren, and he's self-experimenting with the cell-penetrating peptide FOX04-DRI, which I don't know if you're familiar with these mouse studies that had dramatic results, regrowing hair and youthful appearance. So I was curious what you think about people who self-experiment. Is that a good thing? Is that a bad thing? If you're 70 years old, you're going to be dying soon, or 80 years old, you think, well, maybe should I try this?
So
this
you're
particular
asking
drug?
whether we're going to publicly suggest that people self-experiment?
Yes. See if anyone's brave enough, but that's all I had. So those two questions.
So who wants to chime in on that? And by the way, the public, Vijay doesn't have to do that. Yes, right? I'm going to say it, right? You may kill yourself. It may be the stupidest thing you ever do, right? But maybe we'll learn something, right? Yeah, try and make sure if you self-experiment, try and make sure you publish well, maybe film it, video it, right? Don't sue me for it, because if you blow out a kidney or something, not my responsibility, your responsibility, figure it out. But I think, I'm a proponent of self-experimentation. I know some of you may be or may not want to publicly say that. But go ahead, Tom. It sounds like you had a thought there.
No, I get asked this all the time, what are you taking? Actually, I don't take it, but I do, we were talking about this before, I do intermittent fasting. That's a form of self-experimentation, but I don't take any of the, the current drugs that are on they're in the sphere of what might work. Yeah.
We're not talking about nutraceuticals, right? We're talking
Correct.
about actual interventions like medication.
But I would make just one point. Other people have, when we first published the parabiosis work, I received, the most calls were from men's health magazines and cosmetics companies. And rightly or wrongly, there's a lot of money in that industry to fund research on aging, and ideally if some of that could be channeled to, even if it is cosmetically driven about skin aging, there's a lot of opportunity there to get basic biology. But that's where the money is.
Hi. We were talking about the challenge about funding, which is always a challenge, but I would like some takes on the challenge of data sharing, because we're talking about biomarkers. At the end of the day, when we talk about biomarkers, we're talking about data. I personally work with data in the anti-aging space in skin, and there's actually, when you go outside, you see there's a few data actually available for you to work and actually use the new tools we have for trying to figure out things. So with few data, we have been able to do really good advances in our company. I would like to think about, what do you think about how the industry should come together to actually share this data and try to work it out?
I'll comment on that, because I do have strong feelings about that, because I was incredibly frustrated as a postdoc working on a whole genome sequencing project, knowing that in the world tens of thousands of genomes had been sequenced, but having access to about 100 of them in the public domain. And that slows things down tremendously. So I think that I was really excited to read about the UK Biobanks initiative, where they're going to release 500,000 in the next few years. It's wonderful. I think that really is the way the field needs to move, because for some of these things, you really need, the sample size really does matter. For other ones, it might not, like if you're looking for a few strong factors that change in parabiosis. But if it's a matter of looking for a rare mutation, then it's all about the sample size. And I think that it really slows down discovery to not have those initiatives. And you are seeing pharma companies like that. That's why that partnership was so exciting, right? Because Regeneron is on board. These pharma are willing to embrace that model where they'll have sort of private access to the data for a year and then open it up to a wider community. That's right. Yeah, the algorithms, you can only do very simple things, right? Fit very simple models with that many samples. And then the sort of space of models and the ability, the predictive power really increases substantially. So yeah, there's only a few biological data types that I think are there yet, but what you can do with them is better.
Yeah, one of the things I think is interesting is I think a lot of people just don't want to give the data away, and I can understand that. But federated learning, where you don't give the data but the algorithms run, that's actually a possibility, and actually even cryptocurrencies that are doing interesting things there, that could be the future of how to have it both ways.
All right, so we're in the midst of JPM, which is a crazy health conference that most of us probably know about, that's crushing our lives and creating meeting after meeting after meeting. So I go around and I'm asking people about the aging biology thing because it's a biology conference. One meme that keeps popping up, I want to ask you to comment on, so I'm bringing it into a little bit more of a science direction. As our cells age, the DNA in them that's the master control starts developing, accumulating more and more mutations. How much of an upper bound do you think overall somatic DNA mutation places on the kinds of interventions that we could do to extend lifespan?
Well, in the used car model, I guess you can have a tissue that's chock full of mutations, but you can just swap it out for a new organ. So of course it depends on the intervention. Or if you can selectively target those populations of cells that are damaged, that's entirely reasonable as well. So I think there are ways around that, but it is going to sort of upper bound what you can do.
Yeah, in the organ model, you could introduce new mutations that are not deleterious, but actually that are helpful. So, maybe someone here has a fantastic kidney that we'd all like to have, something like that. So, I don't know, I mean, this is science fiction, but I think it's a natural direction to go.
I think there are also really cool things that we've actually chatted about in the past. So one of the things that I used to be super curious about was why does the brain, which doesn't have a lot of dividing cells, somehow manage to stay relatively healthy over an extremely long period of life? And one interesting fact, or that there's one paper about it, is if you take, for example, mice neurons, and you transplant them into rat brains, they can actually survive healthily for longer than a natural mouse lifespan. So there's some way in which neurons are able to continue to be healthy for longer than you'd expect them to naturally have to be. One thing that a postdoc recently, a friend recently mentioned was the fact that neurons can divide and kind of push off huge globules of junk, which seemed like in worms at least an interesting mechanism. I'm not sure that's been shown in people at least, but I think just that whole space of how the heck do neurons survive for so long is a super interesting one to study in this respect.
Hello, everyone. Hi. So I think you didn't mention senescent cell removal, and I would like to hear your thought about that and how close it is to market. And then another thing you didn't mention is going with evolution and talking about what you said about driving in old cars is the way you drive old cars, maintenance and getting new parts. So the work that eGenesis is doing to try to get new parts for humans in pigs, and then what about cloning, for example, that you can get new parts for humans?
Sorry, what's the question though? I mean, that definitely.
So I think senescence first. So getting rid of senescent cells. How close is that to market?
Okay, sure. Sorry, I don't want to talk too much. So I think one of the exciting things here was, so for those who don't know, a senescent cell is a cell that, as you get older, starts to secrete pro-inflammatory factors, refuses to kill itself, and eventually might help to aggravate a lot of the pathologies that occur with aging. One of the fascinating papers that came out in this field was in 2011, where people at the Mayo Clinic got rid of a lot of senescent cells in a progeric, that is accelerated aged mouse model, and showed that it stayed healthier for longer. And I think one of the interesting things was my prediction from that was that you couldn't actually impact lifespan, that was just sort of a aging pathology thing. But last year, a paper came out showing that actually getting rid of senescent cells in a normal, healthy, wild-type mouse does increase mean lifespan by about 30%. This is pretty fascinating, and I think actually kind of a large step forward for the field. There are a lot of different approaches to targeting senescence, and we're definitely invested in at least one of them right now. And they're expected to hit the clinic in the next couple of years. So when people ask, what's interesting in aging biology, there are things that are already in the clinic that you might genetically expect to impact lifespan. But in terms of things that were motivated by the idea of impacting aging itself, probabilistic analytics are some of the first things that will be hitting the clinic to answer or at least address that question.
I mean, maybe as a general answer or comment to the other one, I mean, I think that's an intriguing direction in terms of organs. You know, I think we should not be naive to think that this whole space is immune to the Gartner hype cycle, and that, you know, to think about where we are, and to always think that there will be the crash coming where, you know, who knows, maybe you have transgenic pig organs and it turns out that's not a great idea. You know, and then the question is how to survive long enough so that you can get past the crash, and it becomes an issue of longevity, I guess, but and to be able to get to the other side. That's very much on my mind in terms of both how to push the field forward and obviously how to push individual companies forward.
So dovetailing off the mutation accumulation question, I'm thinking back to Tom's comment from the beginning about extending healthy lifespan versus maximal lifespan. And in my mind, I'm thinking about that paper, I think out of Jan Vijk's lab from a year ago about like potential upper bound essentially on human lifespan. And I'm just wondering what the panel's thoughts are on even the feasibility of extending max lifespan at all biologically humans, or should we solely be targeting extending healthy life?
I mean, the assumptions made in that model are sort of, it's under the assumption that you don't do anything, right? So
Yeah, it's a pretty controversial paper. I was just wondering.
So I guess that the maximum human lifespan we've seen before is not anywhere close to a true maximum. That said, I mean, there are some cell types that you can't replace, like the brain, which lasts a lot longer, but eventually the DNA damage probably does pile up, right? So what would that look like? I don't know, but that's probably the more true upper bound we have right now. Or,
I would say that is, I mean, so I'm looking out, right, and everyone here is sitting down, and you're like, this is one panel that maybe you'll go to, and it's kind of like, okay, this is interesting, but like, it's super, super weird that in mice you can change a single gene and get an increase in maximum lifespan. Like, that is so counterintuitive. You would not expect that to be the case. So either that completely fails to translate over to humans entirely, and the mice and humans are just so different, there's no overlap, or there's pretty good evidence that we'll be able to impact maximum lifespan. And that's something we'll be testing like in the next couple of years, like the next literally like 2 to 3, which just blows my mind. Like that's crazy.
Actually, does that go straight to humans or it goes to primates? I mean, what's the next test?
So, great point. Unless Kristen can successfully create a biomarker in the next couple of years that with 100% confidence predicts maximum lifespan, we will not know sort of from that perspective, but we will start to have therapies based on the assumption that this is correct, being tested for age-related diseases. And so if they're efficacious where others weren't, that'd probably be a pretty good proof of concept that this general mechanism is one worth pursuing.
Question, so thank you guys for all the work that you do. Giving some context, so I mean a lot of people think about biotechnology as the age of computers back in the day. And do you think, 2 questions, do you think are we, from a product perspective, are we at an age of computers where the technological innovation and the understanding of disease is accelerating at such a rapid pace that that now is the time to invest as opposed to the time for government to invest, which is, you know, early stage. Now is the time for commercial people to invest because we are at that dawn. And the second question is for aging. You know, Joe Betz-LaCroix with Health Extension has talked about, you know, and Kristen, you've mentioned this as far as characterizing aging as a disease. And, you know, thinking about a downturn. One of the things that propels, you know, investment is successful outcomes. And, you know, what are the efforts to, I guess, say aging is a disease rather than finding these surrogates, or maybe surrogate markers is really the answer, Kristen, to then make a market easier to reach from a commercialization perspective?
Yeah, maybe I'll start. So, I mean, in terms of what's different now, a company like a startup needs a product, right? And so that pivot point from basic research into a company is when you actually can sell something and you can make something. And I think there's many examples of that now, so I think that's what's different. You know, it would be natural to think about aging as a disease, but I think that's a shift that will take some time, and actually that'd be a fun thing to talk to our friends in DC about what that would be. But until then, there's plenty of indications that connect with aging that we've referred to that I think are great surrogates to start off with. And so I don't think that's the major barrier.
So I guess I would make one point, and I don't see aging as being characterized as a disease anytime soon. Having said that, if you take any one part of aging, that can be characterized as a disease. So let me give you a perfect example. I mean, the problem, again, in a way, is measurement. Because you take a disease like osteoporosis, it used to not be a disease. It used to be what happened when you got old. And then there were a drug to treat it, and you started to measure it, and it became a disease. So I see aging as being this complex array of things that change, each of which will become a disease. But I don't see us actually as a society saying, okay, we're going to characterize this age as a disease, and we're going to then use that as a readout, and the FDA is going to say yes. I really think it's going to be, sarcopenia is a perfect example. You lose muscle as you get older. We don't call that a disease. We will. It's for the exact same reason. We'll find something to treat it, and we'll find a way to measure it. And then so I think, skin aging, muscle, so that's how I would think is really the near-term future of that.
Yeah, I'll comment on that because I do think it's pretty recently where we've been able to study human aging, right? Like the reason why all these innovations were made, all these discoveries were made in invertebrates in the 1990s was because they lived for 2 weeks, right? And how do you measure human aging? You can intervene and sort of look at the phenotype, which is how long you live, but that takes decades. But now we have new technologies and new ways to analyze them to sort of look at aging in a human. So you can look cross-sectionally, you can look at human centenarians versus regular people, and you can measure everything. You can measure hundreds of thousands of variables and learn from the data what factors might be causal there. And that's a fairly new ability that we haven't had for very long. So I think we're sort of really just starting to map out the most important pathways in human aging, which aren't necessarily the same ones in invertebrates or even in mice.
Yeah, I mean, genomics, proteomics plus machine learning is a very powerful combination.
Exactly.
So we have time for one more question. So, okay, so we'll do two more questions, two quick fires. So, and then if you can just pass it back.
Okay, so this is intentionally simplistic. So, when do humans start aging?
At conception.
Yeah, sure.
We have agreement on that?
I think that if you were to characterize, there would be superimposed, well, this is just my opinion. So that if you were to superimpose development and aging, it would be an overlapping, so you can't see aging for much of development because so many things are changing toward growth and growth, I would say, growth and proliferation. But it would be very hard to date back the beginning of aging to something like, reproductive maturity, or because if we get at this issue of, what is aging fundamentally, we still can argue about this, but if it's mutation accumulation, it will be happening from conception. And so I would say, I said it a little bit facetiously, but certainly the processes, I would say, are starting way, way before you might think they are, and potentially all the way to conception. So I would love to, if anyone wants to.
I mean, other than the potential philosophical questions of when an embryo is a human being, I think I completely agree that it's just one continuous process.
There's another way to answer, which is at what point in time would you have to intervene in order to extend lifespan by the same amount as if you started earlier? And so for something like CR, for example, you can hypothetically start later in life, and in fact, it might be worse to start CR as a child than if you were to wait until your mid-20s, which is one of the reasons why I've never bothered with any kind of dietary regimen. But yeah, there's a lot of evidence that for some interventions, at least starting later in life is equivalent to or better than starting earlier, which could be an interesting spin on the question.
So would that be like a middle age?
Right, exactly. Like maybe taking metformin after your 40s. Although, no clue.
Yeah, and part of that is because, right, we are aging from conception, but there are these inflection points, right, where it starts to get an awful lot worse year over year, and that's when you want to.
Yeah, tell me about it.
I wonder if the panel can expand upon what Tom touched upon earlier, that is the societal implications, the ethical implications of the aging research, of creating a larger population of a geriatric population. Because we are already running out of young people to pay for the Obamacare.
Yeah, we can have old people pay for Obamacare. I mean, that's the idea, right?
I mean, just briefly, but I think this is all about creating a less geriatric population. Like right now, you get your first age-related disease when you're maybe in your 60s, and then you have it chronically for a decade or more. But these interventions that can work in mice delay the onset of all of these diseases, right? So you actually end up sicker for a shorter period of your life. That's the dream, at least, and that's what we can do in mouse models, and we're hoping that translates into humans.
One last question.
It's a comment. Tom, you're very good at prediction. So due to a lot of lobbying, because people are working in this space, the ICD-10 code M62.84 was approved October 1st of last year, 2017, for sarcopenia.
So, we're gonna squeeze in one more. Okay, one more question, and then that's it. That's it. All right.
Hey there. I just wanted to ask a question about the other kind of half of the technology angle. When it comes to actually doing research in the lab, are there any technological capabilities that you see researchers asking for that they currently don't have that would really enable the rate of research to increase? Does that make sense? Or?
There are so many. I mean, so let me just give one example. So, a lot of people are interested in proteins, in the blood, right? So, or in a tissue, and measuring proteins. So, sequencing DNA is a commodity. You can send it out, you get it, and it's exactly right, and it's cheap. Understanding proteins, what's there, how much they are, how different they are, we're far, far from that. So that's just one of many examples of, technology that's evolving. That if we had the ability to identify and measure and characterize proteins the way we do with transcripts, it would just vastly increase. So that's just one example, but I could give you dozens more that every day in fact, I would say, obviously, scientific advances occur because of findings and conceptual advances, but few things accelerate more than technological advances. And so every day, I would say every experiment we do, we think about, well, wouldn't it be great if, and I just give you one example. So
a huge thank you to our panelists. So huge thank you.